Professor Pollock. This is James, and this is my endocrine systems integration final presentation. So, starting off with hormones, um, as you'll see throughout the slides here, all the citations for all the information for each slide deck will be on right below the title. But so, beginning with hormones, they're the chemical messengers of the endocrine system. Um, There are some classes and different characterizations of hormones. You've got messenger types, classes, their chemical composition, and then their mechanism of action. Within the messenger, you have the first messenger, which is the hormone itself. And then the second messenger is somewhat dependent on the mechanism of action of the hormone. Um, Hormones differentiate in between the way they initiate. Um, Either transcription at the nucleus or initiate cascades to the cytoplasm via membrane receptors. It just is dependent on um, the type of hormone that it is, and we'll get into that further in a bit. Classes of hormones. You have endocrine, which is specific to the endocrine system. It just means that hormones that are synthesized and secreted move throughout the systemic system to target tissue that is away from where it's secreted. Whereas paracrine is um, hormones that are secreted uh, from their tissue or sites and they act on neighboring cells. Uh, Example of paracrine would be like maybe not necessarily hormone, but inflammation cells like interleukin 1 or tumor necrosis factor alpha. These can be released um, from immune cells and they act on tissues near them. And then autocrine is a class of hormone where it secretes from the gland or the tissue, and the hormone acts on itself. An example of that would be insulin. It secretes from the pancreas beta cells, and then it binds to those beta cells as well. Chemically, the classification of steroids, or I'm sorry, not steroids, of hormones. Chemically, you have steroids, peptides, and then amino acid derivatives. And that'll be covered shortly here. And then mechanism of action, it's not necessarily lipophilic or hydrophilic as a mechanism of action. Those are characteristics of hormones that somewhat determine their mechanism of action. So we have genomic and non-genomic, right? These are the true mechanism of actions as steroids are lipophilic, but steroids are genomic typically because they will act directly on the nucleus. They have receptors in the cytoplasm that kind of shoot straight to the nucleus. Because steroids are lipophilic, they can bypass the plasma membrane or the cell membrane and go straight to the cytoplasm to their nuclear reactors, the receptors, and do their heat shock protein, all their cascading. Whereas peptides are more hydrophilic, and those sequences are non-genomic. And we'll cover all that in a bit, but um, really lipophilic and hydrophilic kind of help describe a mechanism of action just based on how the hormone operates. All right, so steroids, they are synthesized from cholesterol. Again, they're lipophilic. Don't worry about these arrows here. It's not necessarily meant to demonstrate a progression, but they can diffuse through the membrane. They have cytoplasmic or nucleic binding, right? It just kind of depends. It's kind of one and the same. Not one and the same, because obviously, if it's within the cytoplasm, that's not exactly the nucleus, but some steroids, they start their binding in the cytoplasm and then it moves into the nucleus onto the DNA to transcript mRNA. Sorry. Some examples of steroids are estrogen, we have androgens, aldosterone, progestins, Um, an important uh, characteristic of steroids is because they're lipophilic, they require a carrier protein to move throughout the system. And by system, I mean systemic circulation. Um, so you have albumin. Albumin is a big one. It carries good amounts. Uh, uh, man, sex hormone binding globulin, cortisol. Can't remember what cortisol, but there's all steroids will require some sort of carrier protein to ensure their movement. Peptides, on the other hand, they're different than steroids because they are comprised of amino acids typically ranging from 3 to 200, depending on the chain. These are water soluble, right? So they're hydrophilic. They require no carrier protein. They can move freely through blood plasma, um, but they must bind via receptors at the membrane. They typically do not have genomic properties binding straight to the nucleus. They initiate pathways throughout the cytoplasm via their binding at the membrane that start a cascade of events in the cytoplasm, depending on the type of um, uh, pathway that's used within the cell. Um, some types some types of peptides. We have oxytocin, vasopressin, but those are specifically posterior pituitary hormones. Calcitonin from the thyroid, parathyroid hormone, renin. Um, Insulin is a peptide, growth hormone, adrenocorticotropin hormone, follicle stimulating hormone, luteinizing hormone, uh, thyrotropin stimulating hormone. I'm sorry, thyroid stimulating hormone. These are all examples of it. An important point to differentiate between steroids and hormone peptides. Um, Steroids, because they can. Let's stick with peptides. Peptides, because they can bind to the membrane, uh, they can they can kickstart changes within a cell within seconds to minutes. Whereas steroids require more nucleic transcription and changes, right? That can take more, still like longer minutes or hours. It just depends on the process or what the uh, hormone is telling the cell to do. So amino acid derivatives, they are derived from amino acids, but primarily tyrosine and tryptophan. Tryptophan, just quickly, is uh, the derivative for, or it leads to melatonin, melatonin, and it helps with your sleep cycle from the pineal gland. Not covered here too much, but that is that amino acid derivative for that hormone. Whereas tyrosine specifically is for catecholamines, so epinephrine and norepinephrine, uh, and our thyroid hormones. So that's a crazy Y, thyroxine, and then well, let me go back to that. That's T4, and then try iodothyronine. That's T3. These are our main tyrosine derivatives. Um, and then amino acid derivatives can be either hydrophilic or lipophilic. It honestly just depends, right? Catecholamines bind at the um, membrane to initiate cell response within seconds, right? Think of our fight or fight. That's an immediate response, mobilize energy. But thyroxine and we'll just say T3 and T4, right? They can have both properties. They can be hydrophilic or lipophilic and then have genomic properties where they're changing nucleic transcription uh, over hours to initiate some sort of cellular metabolic response. So again, I know it's kind of already covered genomic and non-genomic, right? This is going to have nucleic responses, right? And that can take hours, you know, or like longer minutes if need be, depending on what it is. And then non-genomic membrane response, which leads to a cascade of uh, cytoplasmic, losing the word, cytoplasmic responses, just depending on the hormone. Some pathways for that non-genomic, we have the mitogen activated protein kinase pathway. We have the PI3K or phosphodienosytide 3 kinase pathway. We got the cyclic AMP pathway, which kind of correlates with adenyl adenylase. Sorry if I butchered that. Adenylase cyclase pathway, and then some ion channel changes. These are all non-genomic pathways that can be influenced by mostly peptides, but some steroids can have non-genomic responses. So here's an example of cortisol. So this is cortisol specific, right? This is the chemical structure of cortisol. But here's an example of kind of what I've been saying with genomic and non-genomic. So these two here are genomic. Oh, sorry. It was non-genomic, and then this here, genomic. You can see the genomic, right? Which is it's this yellow. This yellow is the membrane. It can bypass straight through the membrane and go to its cytoplasmic receptor, right? In this case, it's intracellular IGR intracellular glucocorticoid receptor, right? Binds to its receptor. It goes through, going through. We'll get into that a little more in a little bit, but it goes straight to the nucleus, right? Binds to the DNA, spits out some RNA that moves to protein synthesis to tell the cell what to do, right? Whereas non-genomic, similar responses, but this will be a lot quicker because we've got a G protein coupled receptor. I'm sorry, it's our membrane glucocorticoid receptor. Start here. These are tyrosine derivatives as well. Um, and we've got our G protein coupled down here, receptor down here. And then depending on what we'll get into it a little bit further in a second, but depending on the type of G protein coupled receptor, uh, different signals will happen for different things. So the AC over here, the green is the adenylate cyclase, and that you can see here ATP. Once ATP comes to AC, it's kind of kickstarts that CAMP pathway, and then we kind of meet here in the middle, just protein synthesis, where protein synthesis comes from the mRNA. Just this just tells the cell what to do. Just put tell cell, right? If we want proliferation, survival, apoptosis, motility, depending on what we want from the cell, uh, it'll happen. Here in these little blue and yellow, or sorry, blue and pink, we've got ion channels. So we've got calcium channel here, potassium channel here. And these membrane receptors can modulate these ion channels, these efflux, influx, whatever, again, whatever the cell or hormone is telling the cell to do. Same thing over here with this other non-genomic situation for here, right? This one's going to PKC, initiating a different pathway, which again just can inhibit certain actions or plus certain actions. Right. I know it's kind of repeating the same thing, but it's extremely important because the hormone tells the cell what to do. and what needs to be done given the either the imbalance of homeostasis or if there's this kind of leads to an imbalance of homeostasis, but like stress, inflammation, growth, right? Whatever needs to happen with those specific cells, the hormone will tell it. So this kind of just goes a little deeper into the mechanism of actions. If we're looking here, this gives a little bit more specific to the G um, protein coupled receptors. Here, the G and the alpha, that's our main um, G protein coupled receptor, and then these little Y or this little gamma and beta part are the subunits, and the GDP is a guanine diphosphate, and that's it kind of keeps it in the off state. By off state, it's not moving throughout the cytoplasm, initiating the downstream signaling, right? So each sub, not sorry, not each subunit, each G protein coupled receptor has a different function, right? So G alpha here, that stimulates AC. If we go back, right, that stimulates the green here. Um, GAS, that stimulates same thing. Sorry, lost it again. AC here, right? G alpha I will inhibit AC. GAQ, that activates phospholipase C or protein kinase C right here. And then this is a little bit out of the scope of right now, so won't really cover that. And these pathways kind of tell you what occurs, right? You've got cell proliferation from you've got cell growth, motility, and then cell motility. Um, these Y, or I'm sorry, gamma and beta subunits are part of the G protein coupled receptors, right? It kind of tells you over here what's happening with those as well. They're affecting ion channels, PI3K, phospho, I'm sorry, phospholipase C. They're just affecting different areas of the cell as well. And again, we're still looking at a non-genomic sequence here. Uh, this photo shows a great example of steroid hormones, which kind of does both the genomic and non-genomic. Just an example of how steroid can essentially do both. Canonical is the accepted or typical, just put typical action, whereas non-canonical is non-typical. So we would expect steroid hormones, right, to bypass the membrane, interact with their nuclear reactor heat shock protein complex, and then move to the DNA and send out the messenger RNA to be synthesized, the protein, and then initiate the cellular response, right? We expect that. And again, that takes time. We're talking hours. Whereas the non-typical is androgen or steroid, like sex hormones, right? We would not expect them to, I wouldn't say not expect, it's just not typical for them to bypass the membrane or bind to receptors at the membrane and initiate internal cascading. Uh, it's just not often an approach for them because there are different hormones or different signaling mechanisms to initiate these quick responses. Um, so it's just an interesting topic that, and I think this article here goes into the Abikar et al. Article, goes into a little bit deeper than the scope of the class, but it's an interesting topic to really examine, right? The difference of non-canonical signaling for any hormone, either be um, peptides, amino acid derivatives, or steroids. All right, so endocrine reaction. We've got the major axes here between the hypothalamic pituitary adrenal axis, hypothalamic pituitary thyroid axis, hypothalamic pituitary gonadal axis. We've got the growth hormone and IGF-1 axis, which still utilizes HP or hypothalamic pituitary. And I'll cover a little bit more in the following um, segments, but all these axes are correlated and finely tuned via negative feedback mechanisms, uh, with the exception of the HPO, hypothalamic pituitary and ovary, which utilizes a positive feedback during ovulation. But they use these feedback mechanisms to maintain and ensure homeostasis, and it's tightly regulated. Issues occur, which I might be a little ahead of myself, but issues occur when feedback mechanisms canceled or disrupted via, different types of whether it be inflammation or some sort of disease or syndrome, and the endocrine system or whichever axis we're talking loses control of its signaling. Getting a little ahead of myself, but that's how the endocrine system kind of maintains control over what messages are sent out and the appropriate response. The hypothalamic, the HPA, we're just going to go with the HPA, right? Starts with CRH at the hypothalamus, and that heads to the anterior pituitary to synthesize ACTH or adrenocorticotropin hormone, which then travels to the adrenal glands to produce cortisol. The HPT has thyrotropin releasing hormone down to the anterior pituitary to produce thyroid stimulating hormone. And then that heads to the thyroids to produce T4 and some T3. T3 is the more active form of the thyroid hormones, but the thyroid um, produces mostly T4. So what happens is the thyroid hormones, let's say T4, uh, goes to peripheral tissues to be deiodinazed, and there's a whole host of things like reverse T3, T2, right? But specifically, if we're looking at the more important forms, it's T3 and T4. The HPG axis um, begins with GnRH, gonadotropin releasing hormone. And then we get, depending on the pulsality of the GnRH release, we have FSH and LH. In men, the pulsality, right, a rapid pulse preference is LH, and then a slower pulse preference is FSH. And women, it's the same occurrence, but women have a special surge mode where prior to ovulation, their estrogen provides a positive feedback mechanism to release more LH, which helps release the egg from the follicle to induce ovulation. But the end result being for women, we have estrogen and progesterone, which put pro G. And then in men, we have testosterone, and then sometimes DHT, dihydrotestosterone. And then men also produce appropriate levels for them of estrogen. And then women also produce appropriate levels of testosterone to ensure that they make estrogen. If we're talking Theta cells, uh, using LH to produce testosterone and then transfer that into the granulosa cells to produce uh, testosterone, or I'm sorry, not testosterone, estrogens, primarily estradiol with the help of FSH. And then men will produce testosterone primarily, little bits of DHT, a little bit of estrogen, but testosterone can be aromatized in adipose tissue to create estrogen in men as well. So there are some peripheral changes there too. GH and IGF. GH begins with um, GR. Oh, sorry, not GRH, RH. Get back to this. Oh, this is better. Better size. G growth hormone releasing hormone, and that goes to the anti-pituitary. Get GH release, and specific for the IGF-1 or insulin-like growth factor release, GH as it travels to the liver will be synthesized, and the liver creates IGF-1. Uh, and this is where the magic happens for the GH, right? IGF-1 is the signaler for growth. So if we look at that, if we look at each axis, right, got growth here, HPG reproduction, metabolism, and then cortisol is the big stress one. Um, but each of these reacts with one another, right? One can really HPA can has like a master control, not really a master control, but cortisol has such a uh, an amazing, I would say, an amazing response to inhibiting so many things in the body to ensure that survival is maintained and there's enough energy for the system. It's truly unique, and I cover that little bit in this next slide. So this just kind of shows you the interaction between all these systems and how they influence each of these, issues down here from metabolism to bone, reproduction, musculoskeletal, immune system, right? We've got cortisol affecting metabolism through increasing blood sugar, energy mobilization for actual or perceived stress, right? Uh, it affects bone by decreasing bone formation through osteoblast inhibition, right? Osteoblasts are forming new bone, but now we're inhibiting that action because of cortisol. Cortisol directly acts, cortisol can, this isn't the only thing it does, but it can directly act on the gonads to suppress sex hormone development, whether that be the ovaries or the testes. uh, Cortisol induces muscle breakdown if it's long enough, not initially, not just the presence of cortisol will, severely um, break down muscle, but in some cases it can, right, to ensure protein is available to for whatever the body needs. And then uh, short-term immune suppression, cortisol has that ability as well. Uh, the thyroid hormones, obviously metabolic. Uh, They're metabolic hormones for every cell in the body. They actually stimulate osteoblast activity, thus stimulating bone formation. In reproduction, they promote the maturation of Sertoli latex cells and in women, follicle growth. Uh, they also stimulate estrogen production as well. For musculoskeletal, they increase mitochondrial activity and protein turnover, and then it promotes lymphocyte proliferation for immune cells. Sex hormones, those promote insulin sensitivity for metabolism, healthy fat storage, testosterone. Again, it's they promote muscle mass growth and fat breakdown, but that's can be under metabolism as well. Estrogen has a nice ability to inhibit bone resorption, um, and then obviously, sex hormones' primary function is reproduction. Testosterone again increases muscle mass. And for immune system, estrogen has the ability to act as an anti-inflammatory, whereas testosterone is more of an immunosuppressor. And then you got your GH IGF pathway, and oh, it's crazy. Uh, Anabolic nutrient uptake, um, muscle protein synthesis, and lipolysis for metabolism. Especially for children, it's uh, growth hormone is important for bone growth and as we get older, bone mineral density maintenance, maturation of Sertoli latex cells and ovaries, protein, muscle protein synthesis, and then for immune cells, it's important for the development and function of immune cells. And importantly, which can kind of go both to, I mean, it can really go to all of these when we're looking at damage, but growth IGF-1 is important for tissue repair. All of these, not only do these axes up here kind of affect all these different situations at the bottom, but they also affect one another. Like I was saying, cortisol can suppress, you know, TH production. It can suppress um, sex hormone production, and then it can even suppress the activities of growth hormone if in the right amount. If there's an off cortisone to growth hormone ratio, right? Because those two are kind of the antithesis of each other. If there's more cortisol, then we're already going to have a delayed response in growth hormone and IGF-1. Thyroid hormones at appropriate levels can actually uh, lead to cortisol clearance, right? So if we're in a hypothyroidic state, we can have slower clearance of cortisol, leading to an increase of cortisol, right? It actually, again, like I said before, stimulates sex hormone production, and then thyroid hormones can actually uh, increase IGF-1 receptor expression, and that's like a nuclear, that's a genomic action. That's not a membrane action or non-genomic action. Sex hormones. Um, Sex hormones are interesting because the estrogen, if we're just going to go two examples real quick, estrogen receptors in the hypothalamus can actually lead to an increase in cortisol, whereas testosterone and the anterior pituitary can um, suppress cortisol. So interesting situation there. And then similar to what I was saying about cortisol suppressing GH release, GH can also do the same thing and suppress cortisol if there are heightened levels of that. So just a lot of interaction and interplay between these axes. Right? Not one dominates fully. I think HPA axis though does have like a it does have a tight lock on a lot of these other axes because it can have a lot more contribute to it, right? Especially when we're talking about exercise and training or just emotional or psychological stress. Those two alone can contribute to a high increase of cortisol production. So, looking at that, we're sticking with growth hormone and IGF-1 and the HPG axis, with an emphasis on overtraining syndrome and recovery implications. I put down here it's an adjacent discussion with HPA axis because overtraining doesn't happen without elevated levels of cortisol. All right, so what's happening? Oh, and we get that elevated cortisol from excessive physiological or psychological stress. It's a huge one here, inadequate recovery, and then in some cases, insufficient caloric intake can add to that because it is throwing off internal homeostasis, leading to another potential issue that I think we'll cover later. And chronic inflammation kind of adds to that as well. Um, in the GH and IGF-1 axis, overtraining leads to a blunted um, growth hormone secretion, which ultimately leads to a decreased IGF-1, and it is a diurnal secretion. So at night is when GH peaks, right? If we're looking at it, I don't remember the exact time, but it's around our normal circadian rhythm of sleep. So around night, later at night, it will increase. And what happens is that inadequate recovery that's kind of hitting you or hitting the athlete or whoever it is, right? They're not sleeping well, they're getting poor sleep quality, poor sleep amounts. Again, this kind of contributes to it. But they're already getting a blinded GH secretion from overtraining, and the lack of sleep just kind of compounds to it. And GH-IGF-1 axis does not normalize with short-term rest. And that's important consideration because it does make it an interesting biomarker to use for determining uh, overtraining because just short amounts of rest, like if you get everything back on track, it's not going to um, get right back on track because typically it kind of is dependent on a hypothalamic and pituitary disorder. Not disorder, but like dysfunction. For the HPG axis, as I said before, like elevated cortisol levels will directly suppress their reproduction due to the or due to the amount of stress that the body's under. And in men, that looks like low testosterone, and in females, you have functional hypothalamic amenorrhea, which right we have a disruption of their menstrual cycle, and then these increased level of cortisol um, decrease kisspeptin regulation of gonadotropin releasing hormone. And for the body, it really the chronic stress and the elevated levels of cortisol and the shifts away from resources from the reproduction is an adaptive response to ensure that survival occurs. Reproduction isn't necessary for survival. And so the body looks to prioritize where energy needs to go. So for the GH-IGF-1 axis and overtraining, Katie Gianni and Cater, um, they looked at hormonal responses to the insulin tolerant test in overtrained athletes. They had 14 overtrained athletes, 25 uh, non-overtrained athletes, and then 12 control groups. Or sorry, 12 control groups, 12 athletes in the control group. The overtraining athletes exhibited significantly lower basal growth hormone levels. So already we're looking at a blunted GH response just without doing anything. And then uh, overtraining athletes showed a significantly reduced growth hormone levels in response to the insulin tolerance test. The insulin, t- the and then just put it down here. The overtraining athletes just demonstrated delayed growth hormone increase following hypoglycemia. So it's two things. The insulin tolerant test is meant to induce hypoglycemia, and hypoglycemia is a strong signal to the body to release growth hormone so that energy can be readily uh, available to the body. Um, but in this case, these athletes did not have that, right? Um, this really highlights that hypothalamic dysfunction that I was saying earlier because let's just go to this. Because if we're looking for a potent stimulator of GH release, which is hypoglycemia, right? And the hypothalamus is the pituitary zone is a master regulator, but the hypothalamus really secretes the hormones to it to tell it what to do. If the hypothalamus is sensing hypoglycemia, but it's having a delayed response to that, then that's likely going to be the reason why growth hormone is not being released appropriately. So that's just one is one issue or potential issue from overtraining and the growth hormone axis, but not fully verified. Obviously, no one was looking at hypothalamic responses specifically because it's so hard to because it's quite it's a deep brain structure, but it just given the logic of how the body operates, we have a somewhat of an understanding of what would we would expect to happen. For the HPG axis and overtraining, Roberts uh, et al., they examined overtraining effects in the male HPG system using several endurance trained men. Overtraining was induced by doubling the previous week's training volume for these men, and they saw their testosterone significantly decrease. These were, I put these in here, but these were at that was at 001 and this one was at 0.001 as well. These p-values were both kind of going for these. Um, the decrease from 93 uh, nanograms per milliliter to 67 nanograms per milliliter with the standard deviation behind that. And then basal cortisol levels significantly increased from 145.7 to 215 nanograms per milliliter, right? And the huge thing, right? Look at this correlation that they found, an inverse relationship between the two, highly correlated at a negative 0.92, right? Extreme correlation between these two variables, and that's just looking at testosterone and cortisol relative to overtraining. Um, both testosterone and cortisol returned to pre-training values 3 months after overtraining subsided. So a substantial length of time if we're talking about athletes and performance, um, especially if you're in season, mid-season, if you're hitting an overtrained athlete toward the end of season, depending on the sport, uh, 3 months is quite a while to have in the off season to maybe recover. But if we can avoid it through proper programming and adequate rest, adequate nutrition, we can avoid this. Um, and what the authors found with sperm count, which they said it didn't affect, they don't presume it affected fertility, but they saw extension, a significant decrease immediately after training and still 3 months after training at 43% and 52%. So while they're still in the same value, like still in the same, what do they say, normal reference range, that's still a significant decrease, right? So, and that's only, I can't remember. I think it was a 3 month training plan, anyways, that they were inducing overtraining. So think of, right, that's you get a 43% reduction and a 52% reduction after, or let's say 43% after 3 months. Presume you have an athlete training over a longer period of time and you've just now induced overtraining for longer periods. And no one can seem to understand why the issue is, but they're still kind of, they have that unexplained performance reduction. And now it just kind of goes even further and further and further. And so you're likely to affect fertility in some way. Uh, Here's another one specific to women. Cho et al. Examined 40 women prior to, during, and after their enrollment in basic combat training in the Korean Army. Training was intensive, but not specifically overtraining, right? They didn't do exactly what Roberts did in their um, previous study. All women had regular menses prior to basic combat training, and blood samples taken at 4 weeks, or I'm sorry, before, at 4 weeks, and at 8 weeks. And menstrual cycle was determined via questionnaires. Cho et al. Found that cortisol significantly had significant changes from baseline to 4 weeks, from 4 weeks to 8 weeks, and then from baseline to 8 weeks. So at every time point, they had significant cortisol changes. Corticotropin releasing hormone had significant changes from baseline to 8 weeks, and then from 4 weeks to 8 weeks. FSH and LH, no significant changes. Estradiol had significant changes from baseline to 4 weeks and stayed at a reduced level. If I'm remembering correctly, it was somewhere around 106 at baseline, uh, picograms per deciliter, and then it, yeah, 40, yeah, it was right there. 44 was the 4 week, and then it went back up to like 52, but it was still significantly low at 8 weeks. And then of the 40 women, 28 had developed irregular menstrual cycles. Uh, so when they did the analysis of all the measurements, they had essentially kind of retroactively looked at it, right? They just had all the women who developed irregular menstrual cycles and who didn't, and then they split them into their own two groups. So 28 were in the irregular menstrual cycle, and then 12 were regular. And then they looked at all that data, and then the differences were not significant enough uh, to say that the differences between the group were significant. Um, and I think this is important. Like the research methods were not indicative of overtraining directly. So, and this is only 2 months. It's a 2 month, not only 2 month, like it's a short time, but it's a 2 month study. And they weren't forced into overtraining. So Uh, I think sure the authors didn't find exactly like over like the training style of the Korean Army, basic combat training induced hormone changes drastically, maybe significantly per the data, but it is evident, given the information, that there were significant changes in hormones. And so, were this to progress another 2 months, would it be significant enough between the two groups? Um, I don't know. I can't say that, right? But I can speculate. It probably would. But it does give evidence that exercise does affect the menstrual cycle if prolonged and done inappropriately. All right. So, getting into endocrine disruptive states, we have low energy availability, which is there's a formula for it, but just to keep it simple, it's exercise. I'm sorry, energy intake, food you eat, versus exercise energy expenditure equals your energy availability. And mathematically, that's less than, let's just say, decrease less than 30 kilocalories per kilogram of free fat mass per day. And kind of similar to what I said about the gonadal HPG axis and overtraining, right? The body's going to prioritize survival over everything. So, how it does that is ensure that the brain has the energy that's needed. And if the brain has the energy that's needed and nothing else really needs it, there's going to be changes in other places, right? The body is going to down regulate metabolism. It's going to slow it down. It's going to suppress your immune system. It's going to stunt your growth development. Hematologically, it's going to reduce your hemoglobin levels. It's going to slow down your gastrointestinal tract, constipation, malabsorption, your heart, hypotensive, bradycardia, endothelial dysfunction, endocrine. We're going to cover that shortly, right? Psychologically, you can develop uh, mental issues or like depression, anxiety. But essentially, that's over the long term. But the brain is going to try and prioritize its survival. Specifically in females, you get this female athlete triad with energy deficiency being an issue, and then you get a reduction in bone mineral density, and you get menstrual dysfunction. Kind of what I was mentioning before. Uh, but that was just specific to exercise. Now we don't have the food available, and so we're getting into this route where one, we don't have chronic stress yet. We just have low energy. But if we introduce chronic stress, cortisol suppressed menstrual cycle, we don't have the food to help kind of sustain uh, tissue repair or tissue function. And so the brain's going to just pack it all in, or it's going to ensure that we have enough energy to survive. Um, so low energy availability kind of bleeds into relative energy deficiency in sport or REDS S, right? And if you look here in this picture, we've got demanding training regimens with persistent energy, increased energy expenditure. So what did I say earlier, right? Energy intake minus energy or exercise energy expenditure, right? This is here. And then on the right side, we've got strict diet regimens, peer related and trainer pressure, persistently low energy intake. This kind of goes over here, right? So now we've got low energy intake, high. My arrows are so bad. Sorry. Let me get that right. Low energy intake, high energy expenditure. And so that's going to leave us with a negative balance of energy availability, right? So what do we have? Energy imbalance. What's that lead? That leads to increased glycogen utilization. It's going to use those reserves. Those get depleted. We have less glucose, less insulin, BCA surge, and then those two, right, combine sustained shift from carbohydrate to fatty acid metabolism, adipose tissue depleted, adipose tissue secretes leptin to tell our body it's an energy regulator, right? And now with lower leptins levels, we have a less of an energy regulator flowing around. So we have impaired energy homeostasis, and the body has to adapt with all of these low energy availability situations. In endocrine, we got activin A decreases, follistatin increases, like we've talked about earlier. Thyroid hormones decrease, growth hormone pulse increases, but then we get a reduction in sensitivity of growth hormone, IGF, and IGFs as well. The GH pulse, right? The body's sensing this energy dysfunction, so it's trying to get more out, but the reduction in sensitivity makes it not used. Catecholamines are the initial response to increase energy mobilization, and then we had an increase in ACTH and increase in cortisol, which long-term stress. Got a reduction in LH, FSH, reduction in sex steroids, functional hypothalamic amenorrhea, male infertility, and then specific to that female athlete triad, decreased bone mineral density, and then whole host of other issues, right? Ultimately leads to this reduction in performance, endurance, strength, negative mental consequences. So in Reds and athletes, Stenquist et al. Examined 13 male cross country skiing athletes in high school, and this was supposed to be a three year study, but it turned into 2.5 years because of COVID, and they did measurements every 6 months. They examined performance, energy availability, body composition, bone mineral density, resting metabolic rate, disorder eating, exercise addiction, VO2 peak, and muscle strength. 38% of these 30% of the um, group had a low lumbar BMD score, bone mineral density score, with a Z score of less than -1. However, they had increases in VO2 peak and muscle strength, but a decreased resting metabolic rate. The authors note that the bone accrual, they had already had low bone mineral density prior to the study, so they think there was some sort of bone mineral accrue or bone accrual issue prior to starting. And the strength improvements are a real unique observation that they found, and I think it leads into another question of the population, right? And then it's important to analyze research methods and so the population being used because this is the only study that I've seen of high school athletes in Reds, at least males. And um, I think just speaking from, I guess, experience or understanding of the endocrine system and growth in general, right? During high school, we have an increase, we're going through puberty, increase in testosterone. Is that increase in testosterone irrespective of the reduction in energy intake? So if the body still has low energy availability and it's still going through this puberty, is that not being affected by the low energy intake? And I just think that's an important thing to consider because otherwise, how are we, this increase in strength, not necessarily how are we getting it? Like it's not something that should be impossible, but it's an interesting find. So I just think that's a critical part when we're looking and critiquing studies. Key et al. They examined 50 road cyclists via sport-specific questionnaire and clinical interview, the CQ, DEXA, and then endocrine blood biomarkers. Data was taken over the course of 2 months. Low energy availability was determined in this questionnaire, which 28% of cyclists had. 44% of the cyclists had low lumbars, BMD scores, Z score less than -1. And then energy availability was the most significant determinant of a low BMD score, Z score. Low energy availability was also associated with total percentage body fat. And then 10 cyclists with chronic energy availability, chronic, it should be an L, chronic low energy availability had lower testosterone compared to others. Um, I thought this article tended to focus on validating this question a little bit more, but I think it seemed to be a valid tool because it likely identified low energy availability. Um, and I think this low, right, we're looking at this number 10. It seems like a low number compared to 50, but I think it emphasizes the importance of individuality and endocrine disruptive states like we understand that. I would say that most of us in this class understand that there's going to be some sort of disruption with low energy availability, but it's not, 100% going to happen to everyone, right? Um, but if we're aware of it and we can understand it, we can educate uh, those around us to ensure that they understand the possibilities of what not eating, uh, excessive exercising, and just a lack of recovery. Reds, again, still we're looking at reds and female athletes this time. And again, kind of highlighting that female athlete triad. I think it's just, I don't want to use the word more detrimental for females, but it's definitely different for females given that female athlete triad and how uh, their women are physiologically set up to one estrogen, high amounts of estrogen, so we're already affecting bone resorption and fat utilization. And so uh, moving, not prioritizing carbohydrates and glycogen and glucose via carbohydrates puts us into a fat metabolism. We're using a lot more fat. We have potentially disordered eating. Now we have lower leptin levels, and then again, we're impacting that energy homeostasis. I'm just gonna say W on this. I don't really know how to say that, but uh, W et al. Examined Reds and endurance athletes compared to non-athletes. We had 23 endurance, 21 control subjects. And they used the leaf Q to determine low energy availability in females. Higher leaf Q scores signified lower energy availability in athletes versus control. 33% of females had amenorrhea. And then compared to controls, leave Q scores less than 8. Energy expenditure was higher in athletes. Compared to athletes with leave Q scores greater than 8, athletes just demonstrated negative energy balance, 90%, with low energy availability of 23 or 24.3 kilocalories per kilogram of fat-free mass per day. Athletes with a leaf score of athletes with a leaf score greater than 8 had lower estradiol, progesterone, and leptin. So, um, it reds is detrimental to both men and female. I don't think there should be a prevalence to get into, one over the other. I think it's just extremely important to educate both groups, both populations, all sports. Um, it's a huge issue. I think should be addressed appropriately. Um, so I've got two reference pages, but if you need them, Professor Pollock, I can just, I mean, you can probably rewrite and see them too. But in summary, the endocrine system is an extremely intricate and like wide-reaching system for the body. Uh, I don't want to say it's more important than any other system, but it is extremely important uh, and specific to the GH and IGF as well as HPG axis. They're extremely important systems that are needed to ensure growth, reproduction, and exercise and performance, but also just you know holistic lifestyle. Um, However, I think it's important to note that these functions, right, a holistic lifestyle, growth, reproduction, exercise, performance, it requires your energy to ensure an anabolic response and the development. Without the adequate energy, the body will uh, prioritize survival over anything else. And I think that is kind of the big story about endocrine disruptive states. So thank you for the opportunity to present this information. If you have any questions, please let me know. Thank